Hello, dear learners and students. Today, I am going to start our second lecture on the biostatistics, that is about the data collection, first step in the field of research. As I have already discussed in the previous section, that data holds a key to any of the research or research procedures, or whenever we are going to start a new project. So all those research projects or research related work are solely and only dependent on our data. Now, it is important that whatever data we are collecting in our field of interest, it should be properly analyzed, interpreted, and reproduced in a presentable manner. So, in this topic, we will discuss that what is data and its importance, and how the different statistical methods which are used to analyze it. So let's start our today's topic. So first, what is data collection? The questions which should any of the research methodology starts with some of the important questions. Unless and until those research questions are not fulfilled or satisfied, we cannot proceed with further work, or we can say further procedures or experiments, whatever we want to do on that field. So it is very necessary that those questions, first of all, it is necessary that why do we need only those data, and what is its importance and how it can help, and what are the different tools and different steps in the data collection methods and procedures, and what are the consideration, what we consider while collecting any of those data. So what is data collection? Data collection is the process of collecting, measuring, analyzing data from various sources to gain insights. Data can be collected through various sources such as social media monitoring, online tracking, surveys, feedback, experiments conducted in the lab, different questionnaires, survey, different type of sensors. So all those are the different sources through which we can collect the data. And here there are different categories also we have given. And basically, what is the source of data? That is either it may be the primary data, which is collected directly from the main source from which to be obtained, or secondary, either it should be some recorded work on that, or some journals, publications, or some previous work done on it. So basically, data is classified on the the two types, that is the primary and the secondary data. Now, data collection methods is different from data collection techniques. So these, first of all, the most important is the data collecting, and then methods, and then what is the technique which is used for the collection of those data. And data collection method refers to a systematic approach to data collection, whereas technique refers to the art of asking, listening, and interpreting. Data collection techniques permit to systematically collect information on people, objects, phenomena, etc., and settings in which they occur. Usually, multiple sources of informations are used because no single source of information can be perfect to provide a comprehensive perspective on the issues being studied. Now, we cannot rely only on a single type of source, or only one source, only one type of method of collecting data. So we have to first of all select those different sources or different modes or methods through which we can collect data throughout for our feedback or for our analysis. So we cannot rely totally on a single source or a method through which we can collect the data. So multiple sources of informations are used. Now here, because no single source can be perfect to provide a comprehensive perspective on the issues being studied. By using a combination of observations, interview, and document review, the researcher is able to validate and cross-react, cross-check the findings. Each type and source of data has strengths and weaknesses. Using a combination of types of data increases the validity as the strengths of one approach can compensate for the weaknesses of another approach. Strategy for data collection generally depends on the research questions and the nature of the variables being investigated. Some of the data collection techniques are given in the following table. You can see here. So the data collection techniques depends on three E's, that is experiencing, inquiring, and examining. Experiencing is through observations and field notes, inquiring through asking, examining through using and making records. So what are the experiencing technique? Here, observation by active participations, privileged active observer and passive observer. Inquiring through asking is definitely through informal interview, structured formal interview, questionnaire, attitude skills. Example is global rating, Likert, semantic differential, focus group discussions, standardized tests, and examining is through archival document, journals, maps, audiovisual tapes, artifacts, and field notes. So in medical statistics, the main source for collection of data is through experiments, surveys, and records. So where number one, experiments and surveys are specially applied to generate data needed for specific purposes, while the records provide ready-made data for routine and continuous information. Experiments are performed in the laboratories of physiology, biochemistry, pharmacology, and clinical pathology, or in the hospital wards for investigations and fundamental research. The data collected with a specific objective by one or more workers are compiled and analyzed. The results are made use of in preparation of dissertations, theses, and scientific papers or publications in scientific journals and books. So experiment is one of the important sources of data collection in the medical field, and it can be used for different type of objective to be fulfilled, especially in dissertation, mainly through dissertation and thesis writing, as well as for our research and journal publications. Next important is the surveys. Surveys are carried out for epidemiological studies in the field by trained teams to find the incidence or prevalence of health or disease situations in communities, such as incidence of malaria or prevalence of leprosy. They are also made to use in operational research, such as assessment of existing conditions and their place to follow a program or to study the merits of different methods adapted to control a disease, such as malaria, or in eradication programs such as smallpox. Thus, they provide useful information on changing trends in health status, morbidity, mortality, nutritional status, or environmental hazards, health practices, etc. Provide feedback which may be expected to modify policy and system itself and lead to redefinition of objectives and provide timely warning of public health hazards. Records are maintained as a routine in registers or books over a long period of time for various purposes, such as for vital statistics, births, marriages, and deaths, and for illnesses in hospitals. Data thus collected are made use of in demography, that is population studies, and public health practices. The data collected are qualitative, obtained by counting the individuals with same attributes such as stillbirths, persons suffering from peptic ulcers, etc. These events are recorded or registered in medical or public health service as a routine. Regular administrative machinery is necessary to record or register the different events. The data collected may be profile or prospective studies at local, state, national, or international level. They are analyzed to assess changes in health or disease situations in the community or population by standard parameters. The statistical data obtained from the above sources can be divided into two broad categories: qualitative data and quantitative data. So qualitative or discrete data, in such data, there is no notion of magnitude or size of the characteristics or attribute as the same cannot be measured. So non-measurable data are the qualitative data. They basically define the characteristics or quality of an object. They are classified by counting the individuals having the same characteristics or attribute and not by measurement. There is only one variable, that is the number of persons and not the characteristics. Persons with same characteristics are counted to form specific groups or classes such as attacked, escaped, died, cured, relieved, vaccinated, males, young, old, treated and not treated, on drug, on placebo, etc. The characteristics such as being attacked by a disease or being treated by a drug is not a measurable variable. Only the frequency of persons treated or diseased varies. By one line of treatment, 20 survive out of 25, while on the other line, 15 may survive out of 25. The characteristic, that is survival, remains the same, but the number of frequency of survival varies. Qualitative data are discrete in nature, such as number of deaths in different years, populations of different towns, persons with different blood groups in a population, and so on. In medical studies, such as data are mostly collected in pharmacology to find the action of a drug, in clinical practice to test the or compare the efficacy of a drug, vaccine, operation or line of treatment, and in demography to find births, deaths, stillbirths, etc. The results thus obtained are expressed as a ratio, proportion, percentage, or a rate. The statistical methods commonly employed in analysis of such data are standard error or proportion and chi-square test, as it will be discussed in later chapters. Now coming to the quantitative or continuous data. So in statistical language, any characteristics, or quality that varies is called variable. In qualitative data, as explained above, the characteristics such as birds, attacked or died do not vary. Only the frequency, that is the number born or attacked, varies. The quantitative data have a magnitude. The characteristics is measured either on an interval or on a ratio scale. In such classification, there are two variables, the characteristics such as height and the frequency, that is the number of persons with the same characteristics and in the same range. Height varies from person to person. It may be 150 centimeter in one and 160 centimeter in another person of the same age and sex. Number of persons with 150 centimeter on the or in the range of 150 to 152 centimeter maybe 10, while those with height 160 centimeter or in the range of 160 to 162 centimeter may be 20. Thus, we find that the characteristics as well as the frequency both vary from person to person as well as from group to group. The quantitative data obtained from characteristics variable are also called as continuous data, as each individual has one measurement from a continuous spectrum or range, such as body temperature from 35 to 40 degrees centigrade, height from 150 to 180 centimeter, pulse rate from 68 to 84 per minute, and so on. The observations or any starts from zero or any starting point in the range which can be measured, which is a measurable data. So the characteristics may be measurable in whole numbers and fractions, such as chest circumstances, 33 centimeter, 34.5, 35.2, 36 centimeter, 37.3 centimeter, and so on. Or it may be measurable or countable in discrete whole numbers only, such as pulse rate, cholesterol, blood pressure, ESR, blood sugar, etc. In medical studies, such statistics are mostly collected in anatomy and physiology, that is in health, to define the normal or to find the limits of deviations from the normal in healthy persons. When the measurement or counting crosses the normal limits, it becomes unusual and may indicate pathology. Some of the statistical methods employed in analysis, such as data, are mean, range, standard deviation, coefficient of variations, and correlation of coefficients. So basically, these are some of the important statistical tools or techniques which are used for analyzing, interpreting, and uh, presenting the data in a study man in a studious manner, or which we can better understand in an understandable manner. So data collected and compiled from experimental work, records, and surveys should be accurate and complete. They must be checked for accuracy and education before processing further. So far, they lie in masses or scattered in the records. In other words, they are mixed and unsorted. Next step, therefore, in sorting or classification of the data into characteristics, group or classes as per age, sex, social class, attacks, etc. They should be presented in such a way that data should become concise without losing the details, arouse interest in the reader, become simple and meaningful to form impressions, need few words to explain, define the problem and suggest a solution to, become helpful in further analysis. For good presentation of data, full labeling, simplicity, and honesty are essential requirements. So, if I conclude today's lecture, I could only say that if best in our daily household activities, if we see that whatever, if when we are going to cook a food, whatever raw materials we have, it is like in the form of data. Now, to eat the food, we have to cook it, add proper spices, salt, pepper, everything in a defined manner or in a rightful manner, so that it not only get cooked but also be more delicious and good in eating. Similarly, is in the case of data collection also. So in the data, whatever raw data we collect, so there are some, we have to include those essential parts which is necessary for our research work and exclude those non-essential parts. Moreover, it helps to make our research, the statistical tools and techniques help simplify our research work. The calculation methods helps to get measurements as well as the detailing of the data from the data, and also helps in to present it in a pre presentable manner, so that the audience should also be able to understand our work, as well also they took interest in the work. So it is very necessary that how we use the data and the techniques to segregate them and utilize them for our research work. So this is was to our today's topic. I hope you would have benefited from today's lecture. Please do like and subscribe my channel Microbiology Tutorials, so that I keep bringing such videos regularly for you. Thank you.